Hello interstellar everyone, welcome to class 12 biology. In the last video, I introduced you to the chapter number 11, biotechnology, its principles and processes. In that video, we learned some basics of genetic engineering and the techniques involved in genetic engineering such as creation of recombinant DNA, gene cloning and gene transfer. Right, remember that particular diagram. That diagram is really important in which it shows all these three techniques involved in genetic engineering. Today, with this particular video, we will start our discussions on tools of recombinant DNA technology. So for recombinant DNA technology, certain tools are required. Right? So those tools are, first of all, restriction enzymes. I told you for the creation of recombinant DNA, we require these important enzymes called as restriction enzymes or restriction endonucleases. And then we also require cloning vectors. Right? We will be talking about plasmids as vectors. I told you about plasmids, right? So plasmids, they act as vectors because they can carry foreign genes, right? So we will talk about vectors. And then in recombinant DNA technology, we require competent host, right? We create that recombinant DNA so that we can introduce that recombinant DNA into some particular organism, right? So that that particular organism becomes genetically modified. So that organism is called as host, and that host should be competent enough to take up the foreign gene. Okay, so the third one is competent host, and then apart from that, we also require certain enzymes like polymerases, ligases, etc. So we will discuss about the first three: restriction enzymes, cloning vectors, and competent host. In this video, we will talk about restriction enzymes. Restriction enzymes are also called as restriction endonucleases. So this particular word is important, restriction endonucleases. Right. So the restriction endonucleases are enzymes belonging to nuclease group of enzymes, and nucleases are those enzymes which can digest nucleic acids like DNA and RNA. Right. So nuclease, they can digest nucleic acids by breaking down phosphodiester bonds present between two nucleotides. You all know that nucleic acids, they are made up of nucleotides. Okay, so two nucleotides are joined together by phosphodiester bonds. So nuclease enzyme, they can break that particular phosphodiester bond to digest or to break apart the nucleic acid, right? And in nuclease, there are two types of enzymes: endonucleases and exonucleases. Endonucleases are those enzymes which can break down the phosphodiester bond right in the middle of polynucleotide chain. So let's say this is a polynucleotide chain, let's say this is a DNA, right? Endonucleases, what they can do is they can break the nucleic acid right in the middle, right? So these are like molecular scissors. They can cut the DNA or RNA right in the middle. So therefore they are called as endonucleases. Whereas exonucleases, they break or they cut the nucleotides from the two ends, either from the 5' end or from the 3' end. Right? So therefore they are called as exonucleases because they are breaking the nucleotides or the phosphodiester bonds at the ends of the polynucleotide chain. So the tool that we use in recombinant DNA technology is restriction endonucleases. So they can break down nucleic acid right in the middle. So they can make cuts right in the middle of the polynucleotide chain. So why is it called restriction endonucleases? Why this particular name, restriction? This is because the enzyme was first discovered in E. coli bacteria in 1963. Inside the E. coli bacteria, this particular enzyme, endonuclease, it acts like immune system of the bacteria. It restricts the growth of bacteriophage. When an E. coli gets attacked by these viruses, the virus injects its DNA inside the E. coli and thereby infects the bacteria and kills the bacteria. Now, when scientists observed the infection of these bacteria by viruses, bacteriophages, they observed that some bacteria, they are resistant to the infection or resistant to bacteriophages. Right? And when they looked inside such bacteria, they found out that there is one very important enzyme called as endonucleases. And these endonucleases, they restrict the growth of bacteriophage within the E. coli. Right? So these endonucleases, they break down the viral DNA and restricts the growth of virus inside the E. coli. And therefore, this endonuclease was called as restriction endonuclease. Now you know why it is called as restriction endonuclease. It's like the immune system of the bacteria. Right? So we obtain this particular important tool of recombinant DNA technology from bacteria. Okay. Let us now look at the names of some restriction endonucleases and try to understand the rules behind nomenclature of these enzymes. Right. So some examples of restriction endonucleases are EcoR1, Hind2, BAMH1, SAL1, PVU2, etc. So these are names of just some of the restriction endonucleases. In your textbook, it is given that we now know more than 900 different restriction endonucleases isolated from about 230 different strains of bacteria. Right. Now, in order to understand the rules or convention behind the nomenclature of these restriction endonucleases, let's take EcoR1 as an example. Right. In the name EcoR1, the letter E is derived from the name of the genus of the bacteria from which the enzyme was isolated. Right. So E over here, it is taken from Escherichia. And the second two letters, it is derived from the name of the species of the bacteria. So coli, in this particular case, CO, it is from coli. And this letter R, it is taken from the name of the strain of the bacteria, RY13. Right? So E. coli RY13. And the last Roman numeral over here, it signifies the order of isolation from the bacterium. So in short, we can say that EcoR1 restriction endonuclease was the first enzyme to be isolated, or the first restriction endonuclease to be isolated from Escherichia coli RY13. So I hope you are clear regarding how these names are given to these restriction endonucleases. Okay, so apart from EcoR1, we have the names of other restriction endonucleases over here. As an assignment, you can try to find out the names of the bacteria from which these restriction endonucleases were isolated. Over here, you have to remember that the first letter is derived from the name of the genus of the bacterium, second two letters from the name of the species of the bacterium, the fourth letter from the name of the strain of the bacterium, and the Roman numeral denotes the order in which the enzyme was isolated from the bacterium. Right. So if you do that, it will help you in understanding the naming convention better. Okay, so you can do that. How does restriction endonucleases cut the DNA molecules? Restriction endonucleases, being enzymes, they are very specific and they always cut DNA molecules at a particular point by recognizing specific sequence of base pairs. That is called as its recognition sequence. Right? The sequence which it recognizes on the DNA molecule where it makes its cut is called as recognition sequence. So it simply means that restriction endonucleases, they don't cut DNA molecules at random locations. Right? They cut DNA at their recognition sequence. The recognition sequences of restriction endonucleases are usually palindromic nucleotide sequences. Now, what is a palindromic nucleotide sequence? In order to understand that, you need to know the word palindrome. So palindrome in English is a word which, when read forward and backwards, they both read the same. For example, this particular word is given in your textbook, Malayalam. If you read it forward and backwards, they will both read the same, Malayalam. Okay, so this is a palindrome. Just like a palindrome, the recognition sequences of restriction endonucleases are palindromic nucleotide sequences. But there is a little bit difference between the recognition sequence and the word palindrome. So in order to understand this particular difference, let us take a look at the recognition sequence of EcoR1. This is the recognition sequence of EcoR1. On the DNA molecule, since DNA is a double-stranded molecule, on the DNA molecule, EcoR1 recognizes this particular sequence, GAA-TTC. And if you look over here, when we read from 5' to 3' direction, this is important. You have to keep the reading orientation same on both the strands. If you read from 5' to 3' direction, the sequence reads the same, GAA-TTC in 5' to 3' direction on this strand, and it also reads the same GAA-TTC on the complementary strand in 5' to 3' direction. Right? So this is called as the palindromic nucleotide sequences. Now there are different ways in which the restriction endonucleases can cut the DNA molecules after finding their recognition sequence. Right? So some restriction endonucleases, they can cut the DNA molecules right in the center of their recognition sequence. And some restriction endonucleases like EcoR1, they can cut the DNA a little bit away from the center of their recognition sequences. For example, EcoR1, after finding its recognition sequence, it makes one cut over here on this particular strand and one cut over here on the other strand. Right. The significance of this way of cutting the DNA is that it will result in two DNA molecules with sticky ends. Right. Now, what do we mean by sticky ends? Over here, you can see that the EcoR1 makes a one cut over here on this particular strand and another cut over here on the other strand. This results in two DNA molecules, two separate DNA molecules, and the two separate DNA molecules, they contain something called sticky ends. Sticky ends are nothing but single-stranded DNA molecule protruding out of double-stranded DNA, right? So this is a single-stranded DNA. Now, they are called as sticky ends because as soon as they come near their complementary DNA strand, they tend to stick to that particular DNA strand. So therefore they are called as sticky ends. Now this is really significant because this has got role in forming recombinant DNA. Right? With the sticky ends, it becomes easier for us to create recombinant DNA. Let us now try to understand the steps involved in the formation of recombinant DNA by using restriction enzymes. In this particular diagram, figure number 11.1, which is given in your textbook, it shows the steps in formation of recombinant DNA by action of restriction enzyme EcoR1. So EcoR1 is the restriction enzyme used in the formation of recombinant DNA. Now, I told you before, recombinant DNA is the DNA molecule which contains DNA from two different organisms. In this diagram also, we have got two different DNA molecules over here which comes from two different organisms. One is the vector DNA, which can come from bacterial, which can be a bacterial plasmid. Another one is a foreign DNA, which can be a human DNA. Now, the important thing to remember over here is that in order to create a recombinant DNA, which means joining of these two DNA, we need to treat these two DNA with the same restriction enzyme. In order to create a recombinant DNA, we have to use the same restriction enzyme on both of these DNA molecules. Why? What is the significance of using the same restriction enzymes on both these DNA molecules? It is because if we use the same restriction enzyme on both of these molecules, it will result in these two DNA molecules which will have complementary sticky ends. The sticky ends of these two molecules, after the EcoR1 makes its cut, it results in these two DNA molecules which will have sticky ends which are complementary to each other. And when the sticky ends are complementary to each other, they will come and bind with each other through formation of hydrogen bond due to base complementarity. And it becomes easier for us to create the recombinant DNA. Right? By using DNA ligase, we can just bind the DNA and form a recombinant DNA. So therefore, we cannot use different restriction enzymes in order to create a recombinant DNA on these two DNA. Right? So we have to use the same restriction enzyme on the two DNA molecules in order to create a recombinant DNA. You have to remember that. Now, I hope that this particular figure number 11.1 is clear with you. Right? You have to remember this particular diagram. You have to practice drawing this diagram. So you can look in your textbook, figure number 11.1, and study this particular diagram. Another important diagram given in your textbook is figure number 11.2, which summarizes the process of recombinant DNA technology. Right? So in this one, we can see over here, there is a foreign DNA over here and a vector DNA over here. It's a plasmid DNA from the bacteria. And these two DNAs, they are treated with the same restriction enzyme. When they are treated with the same restriction enzyme, we can easily bind this foreign DNA to the vector DNA because the sticky ends are complementary to each other, and the ligase will bind and form a recombinant DNA. So this is a recombinant DNA. After the formation of recombinant DNA, now this is carrying a foreign gene. This can be inserted into a host. So in this particular case, E. coli bacteria is the host cell. So inside the host cell, we can introduce this recombinant DNA, and this host cell will now clone the gene. Whenever it replicates, it will clone the gene, foreign gene, and when it divides, when the cell divides, it will pass its gene. It will pass down the foreign gene to its progeny cells. So gene cloning and gene transfer happens after insertion of recombinant DNA into the host cell. Okay, so the important thing over here is also the gene, the foreign gene expresses itself in the host cell. Okay, so the host cell has to express that particular gene. So therefore the host cell is now called as genetically modified organism. Okay, this particular diagram is also important. So you have got two diagrams, this one and this one. So you have to practice drawing these two, and you have to understand what the diagram is trying to convey. You have to understand the concept. Okay, so I hope it is clear. Okay. In the process of creating recombinant DNA in recombinant DNA technology, there is involvement of other techniques as well. One such example is separation and isolation of DNA fragments. Now, DNA fragments are created when we treat a DNA with restriction enzymes. Since restriction enzymes, they can cleave the DNA molecules. Since they are molecular scissors, they can cut the molecules into small fragments. Now, if you look over here, when we treat this foreign DNA with restriction enzyme, the foreign DNA is cut into these small pieces, these small fragments. Now, out of these small DNA fragments, we want only the middle one over here, right? We want this particular DNA fragment because it contains our gene of interest. We want to insert this particular DNA into the vector DNA so that we can form a recombinant DNA. And we want to put that recombinant DNA into E. coli cell or the host cell and transform that host cell into an organism, genetically modified organism, which can express this particular gene of interest. Now, how do we separate this particular gene of interest from the other DNA? Right, that can be done by separation and isolation of DNA fragments, a technique which involves separation and isolation of DNA fragments. We have already discussed about the separation of DNA fragments when we discussed about DNA fingerprinting. I told you DNA fragments can be separated based upon their sizes by a technique called gel electrophoresis. Right, so let's recapitulate. Since DNA, they are negatively charged, when we load the DNA molecules, DNA fragments in this particular well, when we switch on the power source, DNA will move towards the positive end. Right, since DNA are negatively charged, they will move towards the positive end under the influence of electricity, and based upon their size, they will separate on the gel, on the agarose gel. Now, the longer molecules, they will lag behind, and the smaller or the shorter DNA fragments, they will move further. Right, so based upon their size, the DNA, they are getting separated on the gel. Now, after the DNA, they get separated on the gel, they should be stained. Right, the DNA, they won't be visible to our naked eyes. We have to stain the DNA with a special chemical called ethidium bromide. So you have to remember this name, ethidium bromide. After staining with ethidium bromide, we can view the DNA under ultraviolet light, UV light. Under UV light, the DNA bands will appear as orange bands in orange color. Okay, and we cut the DNA out of this gel, right, the gene of interest or the DNA of interest. We cut the DNA out of this gel and we isolate or extract the DNA from the agarose gel, and this is called as elution. Right, so these steps are important. First, we have to separate the DNA fragment by using gel electrophoresis. After that, we stain the DNA and view it under UV light. And after that, we extract the DNA from the agarose gel. So let's say if this particular band contains our gene of interest, we can cut out the gene or cut out the agarose gel and extract the DNA from it. The process is called as elution. Okay, so this you have to remember in recombinant DNA technology. In creating recombinant DNA, we have to use separation and isolation techniques of DNA fragments. Okay, so in the next video, we will look at the other tools of recombinant DNA technology, that are cloning vectors and competent host. So you have to read section 11.2.2 and section 11.2.3 before you watch the next video. Okay, so thank you.